Welcome to our project on the molecular study of the Chaco golden knee tarantula. This presentation is brought to you by NADIAH, NURAMILA, ATHIRAH, AISYAH, IZZATI and ALYA. Our focus is Grammostola pulchripes, the Chaco golden knee tarantula. This tomed ground-dwelling spider is known for its golden knees and impressive size up to 8 inches across. It's native to the open grasslands of Paraguay and Argentina, where it thrives in warm, dry environments. You have just met the Chaco golden knee tarantula, Grammostola pulchripes, a gentle giant of South America. Though not endangered, genetic research helps scientists monitor its health and support conservation. Now, let's explore how molecular techniques help us understand this fascinating species, one gene at a time.
Hidden beneath leaf and soil, the Chaco golden knee tarantula live a life few ever witnessed. But as its habitat faces threat from deforestation to the exotic pet trade, this remarkable invertebrate may one day vanish before we truly understand it. That is where molecular conservation begins at the microscopic level, where science protects not just the animal, but its genetic legacy. Using advanced techniques like in vitro propagation, cloning, and cryopreservation, scientists can preserve the DNA of the rare species. This method helps us breed in captivity without disturbing the wild and safely store genetic material for future research or even reintroduction. In a world where extinction can happen silently, molecular tools give us a voice. Because conserving biodiversity is not just about saving animals, but it is about saving their future, one strand of a DNA at a time.
To protect biodiversity, scientists now look beyond what we see and into the molecular level. In vitro techniques, meaning outside the living body, are lab-based methods that help us study and conserve species using their DNA. For tiny but vital creatures like arthropods, these tools are incredibly valuable. To perform the in vitro techniques, a small sample from the arthropod is collected. The sample is usually a leg or tissue and is taken without harming the organism. One key technique of in vitro is DNA barcoding. Just like a barcode on products, each species has a unique genetic sequence. Scientists extract DNA, amplify a specific gene, usually the COI gene, and compare it to known data. This helps to identify species, discover new ones, and detect invasive or endangered arthropods. Next, we have cryopreservation, which is a method of freezing DNA or cells at extremely low temperatures, usually in liquid nitrogen. This allows long-term storage of genetic material so it can be used for future research, breeding, or even restoration. Another promising method of in vitro technique is cloning. By using a single cell, scientists can recreate an organism in the lab. Though still experimental in arthropods, cloning may help restore endangered species in the future. Together, DNA barcoding, cryopreservation, and cloning show how in vitro science is helping us understand and protect life, even the smallest of it.
In the world of science, even the smallest clue can unlock big discoveries. This is Grammostola pulchripes, the Chaco golden knee tarantula, beautiful bull and often mistaken for its look-alike cousins. To truly know what species we are looking at, scientists turn to a powerful tool called DNA barcoding. It starts with a tiny sample like a shirt skin or part of a leg, carefully collected without harming the tarantula. Inside the sample in the code of life DNA. Using a method called PCR, scientists copy a specific region of the DNA known to be unique in each species. That sequence is then compared to a global database. If it matches Grammostola pulchripes, the ID is confirmed, but we don't stop there. Next, we study genetic diversity, the natural differences in DNA between individuals. Why? Because low diversity means inbreeding, and inbreeding leads to weaker, less healthy tarantulas. Through genetic monitoring, we can protect these animals, making sure the population stays strong and full of life. From pet shops to conservation labs, DNA recording and genetic monitoring are helping us understand, protect, and respect the wonder of nature, one species at a time.
Cryopreservation is a technique used to freeze and store living biological materials in extremely low temperatures. Using liquid nitrogen at -196 degrees Celsius, all biological activity stop, allowing the cell to remain viable for years or even decades. For the Chaco golden knee tarantula, cryopreservation can play an important role in conservation. This species grows slowly, matures late, and reproduce infrequently. Because of that, every individual's genetic material is especially valuable. Cryopreservation allows us to preserve sperms, eggs, and tissue from selected individual. This stored cell become a kind of biological backup, creating a frozen libraries of genetic diversity. In the future, they can be used in captive breeding, scientific studies, or even advanced technologies such as cloning. This gives us a way to protect and potentially recover the species' unique genetic trait. Although cryopreservation takes place in the laboratory, its goal is closely connected to the wild. Each preserved sample contributes to the possibilities that this tarantula will continue to exist not only in storage but in the natural environments where it truly belongs.
Cloning in invertebrates is mostly seen in organisms like echinoderms and ascidians, which can regenerate or reproduce asexually using pluripotent cells. While this shows biological potential, studies like those by Mustard et al. Also helps us understand invertebrate neurobiology better. In terms of synthetic biology, tools such as somatic cell nuclear transfer and induced pluripotent stem cells are being explored for endangered species recovery. Tar cloning also supports large-scale genome editing, which could be useful for future conservation work. However, when it comes to tarantulas, these technologies face major limitations. Cloning isn't yet feasible due to biological and technical barriers. Plus, ethical concerns arise regarding ecological balance, cultural values, and even legal implications. So, while cloning and synthetic biology are promising tools, for now, protecting and restoring natural tarantula population should remain our main focus.
Molecular techniques like DNA barcoding help scientists study genetic diversity and monitor the health of tarantula population. These methods support early detection of environmental threats and improve conservation planning. Continued research and investment in genetic studies of invertebrates will strengthen efforts to protect species like Grammostola pulchripes for the future.